Warm welcome to everyone who's watching this. My name is Sanjeev. I'm 17 and I just finished my pre-university and I'll be studying medicine soon. In this presentation, I will cover most of the ways thrombophilia can be inherited. I've tried my best to make sure that anyone who's seeing this without any prior knowledge in genetics will leave with at least some sort of awareness regarding the topic. You might also learn a thing or two. Stay with me because this is going to get interesting. What exactly is thrombophilia? If you have thrombophilia, it means that your blood can clot much quicker and much easier than the normal person. So if you really look at the picture here, this is the normal person's blood clot and it takes much lesser time to break it down. And if you look at a person who has thrombophilia, the blood clot becomes excessive and it takes much longer time for it to break down. And if you have thrombophilia, you're under risk for DVT and PE. And thrombophilia can be inherited too, which we'll be discussing today. I'll be discussing about four major types of inherited thrombophilia, and let's start off with Factor V Leiden. Factor V Leiden is the most common form of inherited thrombophilia. It is kind of rare in the Asian region, but kind of prevalent in the European origin. Our blood plasma basically consists of 12 clotting factors which help in the hemostasis process or the clotting process. One of the clotting factors, clotting factor 5, a mutation in this clotting factor causes increased resistance to anticoagulant response. What I mean by this is that when a clot forms in the blood vessel, essentially you need a break at some point to stop the clotting process, and the anticoagulant response is responsible for this. When the clotting factor is immune to this anticoagulant response due to the mutation it gets, essentially you're going to have a huge clot, and we don't really want that. So this VTE is going to cause other problems like DVT and PE. We just talked about a mutation in clotting factor 5. So to really understand mutations, I have a simple explanation for anyone that's watching this so they can get a whole idea of what the genetic code is and what mutations are. So every kid's like, playing with Legos and everything. So we give a kid a Lego manual and he makes the Lego car. So to get the manual, you need to get it from a database, printed it out, right? So essentially, in our body, you're going to have the genetic sequence, which I showed you earlier, the coding DNA, undergoes a process called transcription to form mRNA. So it's the messenger RNA, and a molecule known as tRNA reads the mRNA in a process called translation. So after translation, you're going to have the protein. So that's basically the whole central dogma of genetic expression. Basically what I just showed you. The DNA undergoes transcription to form mRNA and translation to form a protein. I won't really get into the detail of each process, but just to summarize, the DNA forms the mRNA through transcription. mRNA is the Lego manual we discussed. It contains the code or the sequence essentially of the protein molecule. And in the process of translation, the tRNA brings the protein in the exact sequence. So we get a polypeptide chain so that the amino acids are linked in the correct manner of a protein. In the translation process, the child, which is the tRNA, reads the mRNA, which is the Lego manual, and brings in amino acids which correspond to each of the codons. So we look at the table here. The amino acids correspond to each of the codon here. So glycine stands for any of these codons, valine stands for any of these codons. So at the end of the translation process, you're going to have a sequence of amino acids which forms a polypeptide chain or a protein. You might be wondering why we're discussing all of this, but just imagine if one of the nucleotide or the letter is replaced with another one. That means the codon is going to change to a completely different one. So you're going to get a completely different amino acid in the polypeptide chain. And what that means is that the protein is going to get modified and it might function abnormally, or you might not even get a protein in the first place and the structure gets destroyed completely. And that's what a mutation is. And mutations can be inherited since the DNA is also inherited to your offsprings. And this is exactly what happens in Factor V Leiden. If you look at chromosome 1 where the Factor V gene is located, a normal Factor V gene would give you a normal Factor V protein which responds to the activated protein C, which leads to the inactivation of Factor V protein. But if you look at the Factor V Leiden gene, there's a type of mutation where a nucleotide is replaced with another one. It's known as a point mutation basically, and you're going to get a completely different amino acid which gives you this whole abnormal protein. And this abnormal protein gains increased resistance towards this activated protein C, which impairs the inactivation of Factor V Leiden, and that leads to enhanced thrombin generation. People with homozygous dominant Factor V Leiden are at 80 times more risk for blood clots. Would hypercoagulable states such as severe COVID-19 increase the risk even more? So let's move on to the next one, which is prothrombin 20210. Thrombin is one of the blood clotting factors, and when it's activated, it becomes thrombin, and thrombin causes fibrinogen. This is basically all the cascade events we're seeing here in hemostasis process. And all we need to know is that in the prothrombin 20210, the mutation of the prothrombin gene, which makes up the prothrombin protein, causes the increased production of prothrombin, which means that you're going to have stickier blood than usual. And when your blood is more stickier, you're going to be more susceptible to blood clots. Now, if you look at the prothrombin gene, it is the same type of mutation, a point substitution mutation. And there's one important thing to note, that there's evidence that the hypocoagulable state is due to the increased efficiency of the polyadenylation site. What they mean by polyadenylation site is that there's a process where the mRNA is converted into a more mature form of RNA, which is known as hnRNA, but we won't really get into that. But a sub-process known as polyadenylation increases the stability of the overall molecule. So an increased efficiency in this site leads to an increased production of prothrombin mRNA. And when we have more mRNA, automatically we're going to have more protein expression. The next type of inherited thrombophilia we'll be discussing about is protein C and protein S deficiency. This is pretty straightforward. Protein C and protein S are some of the natural anticoagulants. As I was talking about earlier, the brake that we need to stop the clotting process, these proteins are responsible for them. So if you don't really have enough of protein C or protein S, you're going to have a thrombosis. And other medical conditions such as kidney disease, which is non-genetic, can cause low levels of protein C2. The inheritance part, I will discuss it in a bit. Coming to the last type of inherited thrombophilia we'll be discussing today, which is antithrombin deficiency. It follows the same concept as protein CNS, as all of them come under the same category, which is anticoagulants. So if you don't have enough antithrombin, just like protein CNS, you're going to have a venous thrombosis. And other medical conditions like kidney disease can cause low levels of antithrombin. You do not have to note that the heterozygous version of the disease is associated with the greatest risk of venous thrombosis. If you give me a few minutes, really, I can try explaining the whole inheritance pattern of all the autosomal dominant diseases which we have discussed today, which is all 4 of them. What we're seeing right now is a pedigree chart, and we use it to study the transmission of genetic disorders in a family tree. And in autosomal disorders, the males and females are equally affected, as the alleles we're talking about are not located on the sex chromosomes. The capital A stands for dominant allele and the small a stands for a recessive allele. An allele is basically a slightly modified version of a gene. And in the case of autosomal dominant disorders, the only condition that's not affected is the homozygous recessive. The heterozygous condition, which is a dominant allele and a recessive allele, are affected, and a homozygous dominant, which are both dominant alleles, are affected. So if you look at this pedigree chart, the father is heterozygous, which means he contains one dominant allele and one recessive allele. And the mother is homozygous recessive. And to simplify this even more, we won't really get into detail, but each gamete of the father is only going to have one allele. So during fertilization, you're going to have random combinations of any of the following alleles just combining with each other, like permutations. So if you look at this picture, it's going to be more simplified. So this is basically the dominant allele and the recessive, and the mother is also unaffected. So a heterozygous parent with another unaffected parent, the baby will have any one of the 4 combination of genes, basically 50% heterozygous and 50% unaffected. So to basically summarize, if one of the parents is heterozygous and the other one is unaffected, you're going to have a 50% chance that your child will be affected with any of the autosomal dominant disorders, which is all of the 4 disorders we have discussed today. But let's say if both of the parents are heterozygous, then what would happen? So if you take a look here, you're going to have basically 4 possibilities. If you look at a Punnett square, on homozygous dominant child, a heterozygous child, another heterozygous child, and a homozygous recessive child. So in the case of autosomal dominant disorders, as I've told you, only the homozygous recessive is normal. So what that means is that 75% all the possibilities of gametes fusing, you're going to have only a 75% chance that your child will be affected if both of the parents are heterozygous. Let's look at some of the preventive measures to reduce the risk of the complications of genetic thrombophilia or thrombophilia in general. As we can't really prevent genetic thrombophilia, we can make healthy lifestyle changes by controlling the risk of venous thrombosis by leading a physically healthy life and by exercising and not becoming obese and not avoiding stretching. and also by stopping smoking completely. And if you have genetic thrombophilia, you should also get medical advice before surgeries and everything. And if you're a woman, you should also get medical advice before taking contraceptive or hormone replacement therapy and becoming pregnant. All of this you should consult with your healthcare professional. And of course, if nobody in your family has a venous thrombosis, the risk is obviously low. And if any of your family members have inherited form of venous thrombosis, you can also take the preventive measures by following them. Thank you so much for giving me your precious time. I try my best to stay as short as possible, but I apologize if I went too far, but that's all I could do. So I hope you understood at least something or the other today and got a lot of information through this presentation. Thank you.